Hello, today we are going to start Ascomycetes, the sac fungi under kingdom fungi. So this Ascomycetes is otherwise called as the sac fungi. So why do we call them a sac fungi? Because the fruiting body is a sac-like structure. The fruiting body, that is the ascus, that is a sac-like structure. So it is called as the sac. Within the sac we find the spores are present, and these spores are called as ascospores. These are called as ascospores, and these ascospores are called as the sexual spores. So due to the presence of this particular spores and due to the presence of this ascus sac-like structure, this fungi is regarded as Ascomycetes. Okay, let's discuss about its general character. So the mycelium present in this Ascomycetes are all of septate hypha. For example, this hypha, they do contains septum in between them. So in septum present, so they make the fungi that is called as uninucleated or multicellular. So these are the nucleus present. So these are the nucleus present and these are the septum present. So when we find septum present and nucleus present, so these are called as septate hypha. Got it? And here we find what it is uninucleated. It is uninucleated and also it is multicellular. Yes, it is multicellular. And the septate condition is mostly found to be present in higher fungi. It is found to be present in higher fungi, but in lower fungi we find aseptate condition, but here we find it is septate condition. So all higher fungi are septate hypha having septum in between them. And one important thing is that in between the septum there contains an opening, and this opening is called a septal pore. So the septum contains that is opening, that is called as septal pores, and the septal pores helps in exchange of materials within the cells or between the cells, that is adjacent cells. Got it? So they have central pore present in the septum, that is called as the septal pore. Now let's discuss about one of the exception case, that is yeast. Yeast is unicellular. So as yeast is unicellular, so they don't have septate, so they don't have hypha, because all the yeast are unicellular in their habitat or in their structure. And mostly Ascomycetes are saprophytic, they are decomposers, they are parasitic, or they are coprophilous, found to be present on dung, or cow dung, or any other waste materials. So saprophytic, they are found on dead organic matter, they decompose the matter. Parasitic, some are disease causing organisms. The Ascomycetes contain septa, and they do contain septal pores, as we have already discussed that in between the septum there is opening present, and this opening is called as the septal pore. It is the character of this particular fungi that is called as Ascomycetes. The pores allows communication and transport between the cells. The cell wall of fungi. For example, suppose you are taking a single cell. We find the cell wall of fungi is normally made up of fungal cellulose. It is made up of fungal cellulose. And this fungal cellulose is called as chitin. So this fungal cellulose is called as chitin. Normally the cell wall of fungi is made up of fungal cellulose, whereas in lower fungi, that is in OUAT, the cell wall is made up of, that is called as cellulose, plant cellulose. Motile structure do not occur in the life cycle of Ascomycetes because they don't have flagella. So no flagella cilia are present. So in the life cycle, no flagella is found to be present. No flagella, no cilia present. So as no flagella, no cilia present, so motile structure is totally absent in this particular group of organism. In majority of Ascomycetes, the mode of asexual reproduction is by means of Conidia. So Conidia is a type of spore produced from a structure, and this is the branch, and from this branch we find small spores are present. Got it? So small spores are present. So this structure is called as Conidiophore. Conidiophore. This is the hyphal structure. This is the hyphal structure. And each conidiophore gives out branching, that is called as the rami. So rami is the branching up. And on the rami we find sterigmata, another branch, that is called as sterigmata. Got it? Upon sterigmata we find, on the sterigmata we find small spores are present, and these spores are called as, that is conidia. These are called as conidia. Got it? And these conidia present, these are the spores that do help in dispersals. Got it? So these spores are the conidia spores, and these are asexual spore. These are asexual spores. So the mode of reproduction in Ascomycetes asexual reproduction is by means of Conidia, and Conidia asexual spores. Got it? And these asexual spores are produced on a stalk-like structure that is called as conidiophore. And the conidiophore contains the first branching of conidiophore, that is called as rami. From rami, a small branchings are present, and these are called as the sterigmata. And from sterigmata, we find the conidia are found to be present. And these conidia, when gets matured, they get dispersed and gives rise to a new individuals. So let's discuss next point. So yes, conidia are branched or unbranched depending upon the nature. So this is how the conidia looks like. These are the vesicles present, these are the conidia present, and here we find sterigmata present, the chain is present, that is called as conidiophore, and we find hyphae is present. So Penicillium and Aspergillus are the best examples of this Ascomycetes that do produce conidia as the mode of asexual reproduction. The female sex organ is specially called as Ascogonium. So the female sex organ of Ascomycetes is called as Ascogonium. And plasmogamy, what is plasmogamy? That is union of protoplasm. Union of protoplasm. So union of protoplasm is called as ascogamy. And this union of protoplasm is by means of gamete-tangential contact. Means suppose this is a hypha present and they have a gamete, and this is another hypha present and they do also have the gamete. So both the gametes are undergoing contact, and this contact nature is called as gamete-tangential contact. And in sexual mode of reproduction, three conditions takes place, that is plasmogamy, that is union of protoplasm, is followed by karyogamy, which is followed by meiosis, This is the process of sexual reproduction in Ascomycetes. And mostly Ascomycetes undergo gamete-tangential contact. And after contact, what happens? They form a common structure. This is contains multinucleated gametes, and from them the fruiting bodies develop. And these fruiting bodies contain spores, and these spores are called as sexual spores. They are called as ascospores, and which differs to give rise to a new individuals. So the best example of gamete-tangential contact is found to be seen in Pyrrhonema, is the best example. And also yeast sow by means of conjugation. Yeast, normally in yeast we find conjugation because in between them a conjugation tube is produced, and this conjugation tube do helps in exchange of gametes, exchange of protoplasms, and by which yeast undergo its reproductions. And in Ascobolus, it is by means of spermatization. For example, there is a female sex organ that is called as Ascogonium. This is Ascogonium. Here the egg is present, and it is present within the hypha. And here the male part is present. These are the hypha present, and this male gives out spermatia. These are called as male gamete. That's called spermatia. And this is the female Ascogonium, that is the egg is present. So these spermatia disperse and gets attached on this and go and fertilize the egg. So this type of fertilization is called as spermatization. And the spermatization mode of fertilization found to be present in Ascobolus. Somatogamy. What is somatogamy? In somatogamy, what happens? Suppose this is one kind of hypha present. This is a type of hypha present. Another hypha gets united with it, and the point of union gets dissolved. So this is what somatogamy takes place, where two different hypha undergo union, and this union is called as what? The somatogamy, and this is found to be present in Pegigia. So this type of union, first the protoplasm will get united, then the nucleus will be united, but suddenly the nucleus never unites. It undergoes some resting stage that is called as dikaryotic phase. We'll study later. And in Morchella, it is autogamy as normal mode of reproduction, as we have seen. But karyogamy is delayed after plasmogamy. In most of the cases, we find the karyogamy is delayed. For example, suppose this is the union of this protoplasm. So here what happened? Plasmogamy is completed. For example, plasmogamy is completed. Got it? So after plasmogamy, we find two types of nucleus present, and there is no union of nucleus. So here what happened? There is no union of nucleus. So nucleus is not united. So when nucleus is not united, this phase is called as dikaryotic phase. Dikaryotic phase. Got it? And this dikaryotic phase is the intermediate phase between plasmogamy and karyogamy. And the nucleus present is called as dikaryon nucleus. Because two different nucleus present, dikaryon nucleus. Got it? So this is called as the dikaryotic nucleus. After that what happens? Now that both the nucleus gets united. So when both the nucleus fused, this is called as karyogamy. Karyogamy. Got it? So karyogamy and plasmogamy, in between them a dikaryotic phase takes place, and this dikaryotic phase is the intermediate stage and is the most advanced character of this particular group, that is called as Ascomycetes. So karyogamy is always delayed after plasmogamy because an intermediate stage is present, that is called as dikaryotic phase is present. So these are different types of reproductions found to be present in this particular fungi. That is, we find planogametic copulation. Planogametic copulation means the copulation that do takes place with the help of flagella. If the gametes containing flagella, it is called as planogamete. Aplanogametic means without flagella. So these are the motile. If the gametes are same, that is called as isogamy. If the gametes are different, it is called as anisogamy. And if one is non-motile, another one is motile, it is called as oogamy. And this type of mode of reproduction is called as gametophageal contact, where the female is called as oogonium and the male is called as antheridium, and they give rise to a conjugation tube or fertilization tube through which the protoplasm and the nucleus gets fused with the oogonium, and this type of reproduction is called as gametophageal contact. And this type of reproduction where both the hyphae gives rise to multinucleated gametophagia, and the union of multinucleated gametophagia is called as gametophageal copulation. So this is also a type of reproduction also found in Ascomycetes. And also we have discussed somatogamy, where two different hyphae union. So when two different hyphae gets united, it's called somatogamy is also found. Also we got that is called as spermatization, where we find Ascogonium is present and the spermatia comes and fuse. So spermatization. So these are different varieties of sexual reproduction found to be present in this particular advanced fungi that is called as Ascomycetes. So let's discuss. So a new transitional phase appears in the life cycle after plasmogamy, that is called as dikaryotic phase, as we have already discussed. So in the dikaryotic phase we find two different nucleus are present, but here first we find what plasmogamy takes place. So this is plasmogamy come, and after that we'll find that both the nucleus will get fused, that is called as karyogamy. This is how in Ascomycetes the reproduction do proceed. So the cells of dikaryotic cells are called as dikaryotic cells. Yes, we have discussed. So each dikaryotic cell's nucleus is called as dikaryotic nucleus. We have already discussed. This form is the shorter phase. The dikaryotic phase is the shorter phase in its life cycle. Very important questions. So plasmogamy followed by dikaryotic phase, which is very short period, and then karyogamy takes place. So normally what happens in dikaryotic mode, if one cells get dikaryon, for example, suppose this is a hypha present. For example, this hypha, one of the cell is dikaryon, but other cells are not dikaryon. Got it? Other cells are not dikaryon. So here what happens, one of the nucleus divide, and after division they transfer the one divided nucleus to the next adjacent cell with the help of Crozier bridge. Crozier, by means of a bridge, and this bridge is called as Crozier. So by means of this bridge, one of the nucleus is transported to the other cell. So the other cell simultaneously develops into dikaryotic cells. So this is how one of the dikaryotic cells converts all the hyphal cells into dikaryotic phase by Crozier method. Yes, by Crozier method. This method is called as Crozier method, bridge method. And some dikaryotic cells functions as Ascus mother cells. Yes, when the dikaryotic cells are present, one is a plus minus a different hypha present nucleus, and they develops into Ascus mother cells. This is called as the Ascus mother cells, and this dikaryotic phase are called as Ascus mother cells. These Ascus mother cells will undergo karyogamy, and after karyogamy they will undergo that is called as meiosis, and under meiosis they will produce the spores, a sac-like structure that is called as Ascospores. So this is how the sexual mode of reproduction is found to be present in this particular group, that is called as Ascomycetes. So normally that converts into asci, the fruiting body asci, and this asci gives rise to Ascus, and Ascus, due to the presence of Ascus, we do call them as Ascomycetes. Ascus is the site of karyogamy and meiosis, as we have discussed now. So here is the Ascus present. Inside Ascus we find dikaryotic mother cells are present. They undergo karyogamy. So they undergo karyogamy. So after undergoing karyogamy, then they undergo meiosis to produce the ascospore. And this type is found to be present in Ascomycetes. So normally we find 4 to 8 types of ascospores are produced. Endogenously means within the ascus. It is not produced by Conidia. Conidia, what happened in Conidia? In Conidia, we find the spores are produced outside. So Conidia are exogenous condition, whereas Ascospores are endogenous. Ascospores are endogenous, whereas Conidia are exogenous. So this is how, this is the Ascus. This is Ascus. Ascus is the fruiting bodies. Within the Ascus, we find the Ascospores are present. Endogenously, these are present, means present within the body, where Conidia are exogenous in nature. And most of the Ascus, we find the two mating hypha are united, because two dikaryotic means they have two different hypha. So with the two different hypha are present, the Ascospores belong 50% of one hypha and 50% other hypha. So this type we find 8 ascospores are present of different strains, either plus, minus of different hypha. Ascospores may be arranged linearly. For example, in Neurospora, we find the ascospores are arranged in linear manner. But in yeast, it is randomly arranged. So this is the condition of yeast and this is the condition of Neurospora. So it has been found that ascospores in Neurospora is arranged linearly and in yeast it is arranged randomly. And the Ascomycetes occurs freely or get aggregated into specific fruiting body called as Ascocarps. Yes, sometimes we find group of Ascus, they are present within a fruiting bodies, and this fruiting body is called as Ascocarp, and these are called as the Ascus. And within the Ascus, we find the Ascospores are found to be present. So the fruiting body of Ascomycetes is called as Ascocarp. Within Ascocarp, we find group of Ascus are present, and these Ascus do contains Ascospores as the sexual spores present. So Ascocarps are of many types. Let's take an example. In Neurospora, they show Perithecium asco, which is a flask-shaped like structure. In Perithecium ascospores, the ascocarp is flask-shaped, and inside we find the ascus are present. Got it? In Cleistothecium, that is in Penicillium, it is closed type. The ascocarp is closed type, and inside ascocarp we find the ascus are present, and inside ascus we find the ascospores are found to be present. It is the condition of Penicillium. In Pleospora, we find it is Ascostroma. It is chambered, means the ascocarp is chambered like structure. The ascocarp is chambered, and within each chamber we find group of ascus are found to be present. And in Pegygia, it is a cup-shaped like structure. In Pegygia, it is a cup-shaped like structure. In Pegygia, cup-shaped like structure, inside cup-shaped, we find the ascus. So these are different examples, valid examples for an exam point of view. Now the fructification of Ascomycetes are edible in some, and in some it is not edible. We can use it. And morels are the most edible Ascomycetes. Morels are type of Ascomycetes which are the most edible form of Ascomycetes. Truffles are also most edible Ascomycetes. And Penicillium is an important character in Ascomycetes because from Penicillium we find medicines have been extracted from it. Penicillium is normally facultative parasites, means they can change their mode from parasite to saprophyte according to the nature of the surroundings. So they are called as facultative parasites, and also they are saprophytic fungi. The mycelium in Penicillium is always branched. The mycelium in Penicillium is always branched. So we are talking about Penicillium general character. The mycelium possesses simple septal pores, as we have already discussed, and each cell is uninucleated and multinucleated depending upon the species, but normally they are uninucleated because they are advanced fungi. So in advanced fungi we find uninucleated because they are mostly septate type. Asexual reproduction by Conidia, as we have discussed, Penicillium reproduced by means of Conidia is very important, and Conidia is a branch. We have already discussed the branching is called as rami. These are the branching rami, Conidiophores. Rami contains stigmata, and in some we find some another structure present, and Conidia is also found to be present. The first branch is called as rami, yes. The second branch is called as stigmata. The second branch is called as stigmata or medullae. The second branch is called as medullae, stigmata, and after that we'll find the that is called present Conidia chains are present. Some medulla are present which are bottle shaped like structure, stigmata. So stigmata is like this structure, like this structure, stigmata this structure, and under stigmata we find medulla, and under medulla we find rami, and under rami we find the conidiophores are present. So these are the arrangement. On stigmata we find the conidias are found to be present. This is how the arrangement found to be present in the Conidia mycelium in that is called as Penicillium, and Conidia are normally uninucleated spores. So Conidia is a normal non-motile because they don't have flagella, so the vectors is air, so they can move through air. So Conidia are dispersed by means of air and germinate to form a new mycelium. This is the condition of Penicillium. Got it? So sexual reproduction, that is in Penicillium, is dikaryotic phase, and they produce ascocarp as the general Ascomycetes do have. And the ascocarp is normally Cleistothecium closed type, as we have already discussed. It is a closed type in Penicillium, and it contains 8 ascospores present in each ascus, and ascospores germinates to form a new mycelium. This is the condition of Penicillium. Now let's move into some important characters or important points about Ascomycetes. Morels. Morels are called a sponge mushroom. The scientific name, other name of morels called as sponge mushroom. It is a type of Ascomycetes, and they are having fleshy sponge like conical cap. And the morels best example is Morchella esculenta and Morchella deliciosa. These are the different form of morels. Truffles are also edibles, as we have discussed here. They are tuber-like structure, tuber-like mushrooms. So they are called as tuber mushrooms, and these are edible, the tuber estivium. In ergot, ergot is a type of Ascomycetes where ergot disease is found in the ears of the mushroom, and the ergot disease contains spores, and these spores are called as sclerotia. So in ergot plant of this Ascomycetes, sclerotia is a type of spores present, and this sclerotia gives rise to a disease that is called as ergot disease. And ergot sclerotia always attacks the cereals group of products, and they causes a disease that is called as ergotism. Got it? And this ergotism contains an alkaloid chemicals that is called as ergotine, and this ergotine will causes abortion in pregnant females if eaten unknowingly. So this ergotism is very dreaded. If unknowingly a pregnant female edible use it, then abortion takes place. So these are used in medicines or abortion medicines. So ergotism is used as a drug to promote expulsion of fetus. Neurospora crassa, this is a type of Ascomycetes which is otherwise called as pink bread mold, is often employed as biochemical genetic work because Neurospora crassa is mostly used in genetic experiment like Drosophila in animal. So Neurospora crassa is sometimes called as the Drosophila of the plant kingdom. For animal kingdom, Drosophila is used as the for animal, and Neurospora is called as the Drosophila of the plant kingdom. Got it? Erysipeae is type of Ascomycetes that causes powdery mildew in most of the products, and this powdery mildew is found to be present in most of the products, and that the best example is called as Erysipeae graminicola. In grams and all, we find this Erysipeae present, disease are present. This fungus, normally some fungus of Penicillium are used for benefits, that ripening of cheese. So ripening of cheese is done by Penicillium. Penicillium roqueforti and Penicillium camemberti. These both penicilliums are used in ripening of cheese. And Aspergillus is a type of Ascomycetes which is called as green smoky mold. So Aspergillus is called as green smoky mold, which contaminates the laboratory products, cultures, foodstuffs, and causes weeds. For which it is called as weed of the laboratory. So Aspergillus is called as the weed of the laboratory. This is the very important question for exam point of view. So Aspergillus is called as the weed of the laboratory. Aspergillus flavus, this Aspergillus flavus is highly poisonous due to alkaloid presence, chemical presence that is called as aflatoxins. So due to the presence of this chemical aflatoxins, this Aspergillus flavus is very highly poisonous. Aspergillus oryza release an enzyme that is called as diastase. That is diastase enzyme is very important enzyme is extracted from this Aspergillus oryzae. This Aspergillus oryzae normally attack the rice group of plants, and this produce an enzyme that is called as diastase, which is extracted for medicinal and industrial uses. Under aerobic conditions and under sugary medium, some of the yeast convert alcoholic beverage or undergo fermentation. So yeast undergo fermentation of sugary solution, carbonated solution to produce alcohol, and it's mostly used in beverage industries or alcoholic industries like beer, wine, cider and toddy. Here we find the yeast are being used. The two most common yeast used by the brewing industry are Saccharomyces cerevisiae, is used in making bread, and Saccharomyces eliposideus is used in making wine. So one is used in, that is, beverage industry, and one is used in baker's yeast. Baker's means we go for, that is, baking, that is producing bread. Gibberellins were the first discovered from the fungi that is called a Gibberella fujikurae, found on the rice, causing the disease Baconian disease of foolish disease. So the foolish seedling disease is caused in the rice plant due to the fungus that is called Gibberella fujikurae. This Gibberella fujikurae, normally what it does, it grows on the rice plant on the growth hormone that is called a gibberellins, for which gibberellin has been, as a plant hormone, has been described or discovered by this help. So Gibberella fujikurae is the perfect stage for the Deuteromycetes growth. So Gibberella fujikurae helps the development of Deuteromycetes that is called as Fusarium moniliforme. And gibberellins, as we all know, are the best plant growth hormones, the natural plant growth hormones. After auxin, we find gibberellin, which is extracted from this particular rice plant, but Gibberella fujikurae never produce gibberellin. No, Gibberella fujikurae, they attack the rice plant to get the edible gibberellins. So from that we came to, Japanese scientists discovered the presence of gibberellin in plant as a growth hormones. Okay, now we have completed our general character. Now let's go from certain practice questions. Assertion: Ascomycetes is called a sac fungi. The sexual pores Ascospores are produced. Yes, both are right, and B is the correct, R is the correct explanation. Got it? Second question: which of the following statement is incorrect? The Mycelium Septate Hyphae is right. Asexual reproduction commonly occur Conidia, right? Motile structure occurs, no. So this is the right answer. C is the right answer. Got it? Third question: Ascomycetes Conidia produced on? No. Yes, they are produced on Conidiophores. The fourth question: which of the following structures is the site of both Karyogamy and Meiosis in Ascomycetes? Ascocarp is the common space. Got it? Or Ascus. Got it? Which of the following fungi belongs to Ascomycetes? The best example of Ascomycetes, that is called as Penicillium. So Penicillium belongs to the group Ascomycetes. Aspergillus and Penicillium are the best examples. Gibberella fujikurae, Morels, Aris Hyphae, everything. There's other examples. Got it? So we have already discussed only 5 questions, but we have ample amount of question per chapter, 1000 plus question per chapter. You can get that ample amount of question from this particular site, or from this you can go for online exam practice from this particular site or online class. If you want direct to get the PDF mind maps, worksheets, MCQs, you can go to this particular sites. Or if you want direct any help or any materials, you can DM this particular site. And also you can get all the materials, PDF notes, everything, MCQs, everything from our Telegram channel. So these are our sources where you can easily extract all the resources. So we have completed Ascomycetes in this class. So in the next class, we'll go for Basidiomycetes. So hope you all enjoyed. Thank you.